what I'm going to be sharing with you is really our work kind of trying to understand how itch is linked to the allergic immune response. Now, itch is a universal sensation. We all know what this feels like. From sharks to bison to seals to birds, we all itch, okay? But what is itch? Well, it was originally defined as an unpleasant sensation that elicits the desire to scratch. But the notion of itch as an unpleasant sensation really belies the torture caused by chronic itch. It's a disorder marked by embarrassment and disability equal to that seen in chronic pain. So if itch can go so wrong, why do we even have it? Well, we know that itch can provide a helpful signal to remove ectoparasites and escape their toxins, things like mosquitoes, things like ticks. But it always seems strange to me that such an evolutionarily conserved process served only one purpose, a purpose that, in my opinion, seems like it could have been achieved with the sensation of pain alone. So if itch really is just a binary symptom with the removal of bugs on one side and disabling chronic itch on the other, is that it? What makes itch more than the scratch? In other words, could there be another role for itch? My laboratory got a clue that there might be more to itch when we examined itch neurons in the skin. Now, here in this picture, you see a zoomed in image of a naive or unimmunized mouse ear. Okay, this is the skin, that thin skin on the mouse. In yellow, you see a web of sensory neurons coursing through the skin. And in purple, you see allergy-inducing dendritic cells. Now, these dendritic cells are the cells that are absolutely required to start up the allergic immune response. Now, although we'd expect these cells to be scattered around the skin, what we see here is that they actually specifically co-localize along the sensory neurons that cause itch. This made us ask if itch is somehow related to starting an allergic immune response. But before we get to the data, maybe I should address the very itchy elephant in the room. Why do we all care about skin responses to allergens? I mean, after all, although we all recognize that allergic diseases affect over 100 million Americans and rising, skin allergies are just one fraction of this. And that's true, but the problem is that the skin is really a major site of sensitization for all allergic diseases, including food allergies. It's poorly appreciated that the majority of peanut allergic individuals will develop symptoms after eating peanuts for the first time. If this is their first time eating peanuts, how did they develop that allergy? Well, there are actually pretty clear and growing data that the skin is a major site of sensitization to food allergens. Exposure to peanut protein and household dust can double your risk of developing a peanut allergy, whereas exposure through the gastrointestinal tract by eating it is actually protective against developing peanut allergies. So this really points to the skin as a special site of sensitization. And to me, that really seems like there is a true need to understand how allergens are recognized in the skin. So based on that picture I showed you before, showing that these sensory neurons and allergy-inducing dendritic cells co-localize in the skin, we hypothesized that sensory neurons played an important role in initiating the immune response to allergens. To investigate this, we first examined the in vivo sensory neuronal response and the dendritic cell response to allergen immunization. We found that a single cutaneous or skin exposure to protease allergens derived from papaya or fungi or dust mites led to an immediate and strong itch response in allergen naive mice that was equivalent to what we saw induced by histamine. And you can see these bars. This was a strong response. And this was the first time that they were ever seeing that allergen. But it was also over very quickly. So it was very transient. It was very immediate. And along with the sensory response, we found that dendritic cells, shown here on the right, were activated and migrated to the draining lymph node to initiate allergic immunity. But what comes first, the sensory response or the immune activation? To answer that, we stimulated sensory neurons or dendritic cells in vitro with allergens. Stimulation of sensory neurons in vitro led to calcium flux that is characteristic of neuronal activation and depolarization. Basically, these peaks on the left show you itch in a dish. However, stimulation of dendritic cells in vitro had absolutely no effect. Together, these data indicate that sensory neurons are the initial detectors of allergens. So it's not just the immune system, it is the sensory nervous system that is initiating that response. But what is the consequence? Is it more than just itch? To figure out if sensory neuron detection of allergens controlled the initiation of the allergic immune response to allergens, we utilize these funny mice called coyote mice to track dendritic cell migration. Coyote mice express a photoconvertible green fluorescent protein. This means that if you shine a purple light on their skin, it changes that green fluorescent protein into a red fluorescent protein. And what's important about that is that allows you to identify red cells anywhere in the body and know that they originated from that site of photoconversion. So it's a great way of identifying cells that started in the skin and ended up somewhere else. Using these mice, we crossed them to mice that we could deplete out sensory neurons from, called TRPV1 DTR mice. And by mixing them together, we were able to see whether or not the depletion of sensory neurons, or TRPV1 positive neurons, led to a loss in dendritic cell migration, okay? And what you see here is that the coyote mice have normal migration of these red dendritic cells, the dendritic cells that started from the skin. But coyote mice that were depleted of their sensory neurons lost that dendritic cell migration. There was no more depletion. They were stuck in the skin, or there was no more migration of dendritic cells, excuse me. They were stuck in the skin. Now, all of this is quantified in the bar graph on the right, where you can see, again, that sensory neuron depleted mice exhibit defects in allergen-induced dendritic cell migration. But how did sensory neurons relay that activation onto dendritic cells? Well, one way neurons can communicate with other cells is through the release of neuropeptides. Now, neuropeptides are these small peptides or small proteins that are stored in preformed vesicles within neurons. So they're available for quick release. Perfect, actually, for immune activation, but also for inducing all kinds of necessary inflammatory effects. Now, two important neuropeptides released by sensory neurons in the skin are CGRP and substance P. Now, interestingly, we found that in vitro papain activation of sensory neurons led not only to that calcium flux or activation that I showed you before, but to the specific release of one neuropeptide called substance P. Now, in fact, not only was substance P released in high concentrations, but it actually was also associated with the downregulation of CGRP release, which is important for us because we know through work from other FASI investigators like like Isaac Chu, that CGRP can actually suppress immune responses. So we were interested by the specific release of substance P and wondered whether or not that substance P was capable of inducing dendritic cell migration and activation in response to allergens. Now, to test this, we went back to our coyote mice, those green fluorescent mice, and asked whether or not direct injection with either substance P or CGRP would lead to dendritic cell migration. So as you can see in these contour plots up top, where close contour lines indicate high numbers of cells, CGRP really had no effect on dendritic cell migration at all. But substance P induced a robust and very specific migration of the allergic skewing dendritic cells, which are marked by CD301b. This is enumerated in those bar graphs below, where CGRP injection had no effect on dendritic cell migration, but substance P injection induced allergen-specific dendritic cell migration that was at least as strong as the allergen itself, but also completely specific for the subset of dendritic cells capable of inducing allergic immunity. So putting all of our data together and also considering other pieces of experiments and other work that I didn't have time to show you today, we found that protease allergens from plant, animal, and fungal sources actually directly activate a subset of sensory neurons that are defined by their expression of TRPV1. Now, this leads to the sensation of itch, as well as the release of the neuropeptide substance P, which acts through its receptor, MRGPRA1, which is specifically expressed by these allergy-inducing dendritic cells to induce the migration. Now, you may be thinking to yourself that this is all well and good, but what does the response to environmental allergens tell us about food allergies? Well, one of the most exciting things to me as a scientist about this finding, about developing, understanding this pathway, is the fact that we have finally identified a cell type that directly responds to allergens. So perhaps we can use this pathway and we can use the fact that sensory neurons respond to allergens to finally understand what makes food allergens allergenic, right? So that's the question, right? That's the question that all of you are wondering, that we're all wondering here at FASI. How do food allergies fit in? How does this pathway inform how we might think about food allergies? Well, anyone who's ever seen their kid or their friend's kid eat knows that skin exposure to food is just part of the experience, right? But food allergens aren't proteases like the environmental allergens we talked about. I mean, some of them are, but let's be honest, most of them are not. But they do share certain characteristics. First, they're heat stable, right? They resist falling apart after cooking. They can still cause allergic reactions even after they've been cooked. They're also resistant to degradation, right? They don't get immediately degraded by all of our enzymes in our mouth and digestive tract. That's how they cause a lot of the problems that they cause. But one thing that we don't often think about is that many of the most immunodominant food allergens are carrier proteins for small molecule ligands. Now, this universe of small molecules in foods, and it is a universe, it is poorly understood and only starting to come to light through work from FASI investigators and others. But what we do know is that these small molecules from both plants and animal food sources are absolutely bioactive. But the question is, could they be allergens? Well, I don't want to get anyone too excited. We don't know the answer just yet. But we do know that by using a neuronal screen of phytochemicals from common food plants, that a shockingly large percentage of those phytochemicals have direct activity on neurons. And what is exciting and interesting to us is that many of these neuronal effects are even stronger than those that are induced by that cysteine protease allergen papain, the one that I showed you all that data for before. And you can see that on this heat map, where the papain neuropeptide release is shown by that, is marked by that asterisk on the heat map. Whereas all those darker areas are phytochemicals with direct activity on neurons. Now, together with FASI, we are working to identify whether these small molecules might actually endow allergic potential to foods, whether or not they might fit directly in this pathway that we've already identified with environmental allergens. Now, in addition to this question, with the help of FASI, we're also interrogating this pathway to understand, well, how it might be altered in some individuals to make them more susceptible to developing allergies. And, getting back to our earliest question, what is it about that itch-scratch cycle that might promote allergies? I hope to share our work in all of these areas with you in the future. But until then, I'd love to answer any questions that you might have. And I'd like to thank you all for your attention and joining us.
Thank you, Carrie. That was a really exciting talk. And it's just fascinating just to think about 10 years ago, we didn't know any of this information, right? And just over the
Yeah.
past few years, you've really been developing this research and seeing this for the first time. And, within the research within FOSSI, we've seen this neuroimmune connection in the lungs, in the gut, and now in the skin. And it's really putting together, all the pieces of the puzzle here of what is really happening. And fascinating to think about that being more nervous than immune as well. But I always love your talks, and everyone loves pictures of animals and babies. So that makes it so nice too. But I'm just gonna please put your questions in the Q&A. Um, and I'm just going to read off a couple of them right now. And one thing that just thinking about through it, when I look at it, is as we think about these small molecules in the skin and that platform that you've developed to look at what initiates this kind of neuroimmune interaction in the skin. How can we think about that from treatment and development of treatments to, either prevent food allergies or, prevent eczema or treat?
Yeah. Yeah. Because, obviously, you know, I, I want to learn about how all of this gets started up. But as a clinician, as a doctor, as a parent, I want to prevent this from happening. I have plenty of business. I don't need any more. So yeah, I mean, I think that that's ultimately the really great question. So what we don't understand, first of all, is how these small molecules, how these allergens are activating the neurons. Okay. And it's important that we start to understand that, to realize, okay, what are the signaling pathways downstream? How can we intelligently really go at this question and think about blocking some of these signaling pathways or blocking some of these pathways downstream? That said, intelligently asking questions and specifically targeting pathways, that works for the development of therapeutics. But a lot of the therapeutics that we still use, a lot of the ones that have been developed based on just screening, right? Of screening for effectiveness in blocking responses, okay? And I think that understanding this fact that these small molecules can directly activate neurons, that we can visualize this itch in a dish, like I like to think of it, we can actually take advantage of that and really think about screening for already established chemicals, for already established molecules, for already established drugs or therapeutics that are out there, to understand, gosh, is there anything that we can use for a different purpose potentially, or can we just really start thinking about completely different models and pathways and drugs, potential drug agents through it by using this screen. So again, thinking of a way to block direct allergen activation of these neurons, but also just to really target it, to prevent that downstream activation. What is really interesting is that all of these itch pathways that are induced by allergens, they're not shared with any other pathway, okay? They're not shared by histamine-induced itch or anything like that, which is a bummer if you're dealing with chronic itch, because that just tells you the complexity of how difficult it's going to be to treat chronic itch. But it really allows us to think about really specific ways to treat allergen-induced itch and to prevent allergen sensitization in people. And, just one other thing, like, the skin, I love the skin because the skin we can access, right? We can access the skin nervous system. We have the ability to think about applying creams and emollients that could, just like they have steroids in them, could have some sorts of therapeutics to prevent this from happening.
Yeah, I mean, we target the nervous system in many diseases, right? I mean, blood pressure being a simple one, right? With the beta
Right.
blocker, to think about that, and so that it does potentially have the opportunity for a lot of different therapies. And, just thinking about my own child when he was first diagnosed and what he was allergic to, he had never eaten before. It was his first exposure to it, and at the time I was like, that's all the food I have in my kitchen, and he had very mild eczema. Actually, he didn't
Mhm.
have much eczema at all, just a little bit.
Yeah.
And so, at the time, they didn't know this connection as much, right? And so
Mhm.
now that we do, what do you recommend for patients who have a child at home, who
Yeah.
has eczema?
Yeah.
And how do you prevent that development of food allergies?
Yeah, I mean, if we knew all the answers, we'd be in a different place. Okay, so I'm going to give you my opinion, and say that potentially there is only data to support some of this opinion. But what I would say is that it's very clear that skin barrier function is incredibly important. So first off, if you have a child that does have eczema, if you have a child that does have atopic dermatitis, really promoting skin barrier function as much as you can through emollient use, through different measures, would be incredibly helpful, I think. The other thing is that we know very clearly now that early oral introduction is incredibly protective against developing food allergies. We know that. But this really brings to mind the need to get that oral introduction and limit the skin exposure to things, okay? And when you're a busy parent, new parent, and you're barely getting a chance to eat, it's totally possible. I remember doing it. You shove down like a peanut butter sandwich, and then you quickly have to change that diaper because you're about to have an explosion. I've been there. But I think, really paying attention to what we are transmitting through skin contact onto our kiddos, and then maybe even sealing in with like diaper creams and emollients. We need to be very conscious about what we have on our hands, versus, what we're feeding.
Yeah, no, it's not easy with, and especially a lot of what I find is a lot of parents are always worried about early introduction at home as
Mhm.
well, which I think may delay it some as well. And I think, getting that advice from the pediatrician as well, that it's okay and they'll be
Yeah.
okay is good too. Um, I'm just going to read some questions here. If we get sensitized through the skin, what could possibly be theories on why there is a higher incidence now and what has changed?
Yeah, yeah. So there are a lot of potential theories here. So number one is really looking at the skin barrier function and whether or not our skin barrier function has changed. We all know that the amount of chemicals in our environment and in our day-to-day living has really gone through the roof. Okay. And when we think about some of these chemicals, we really think about them in such a warm and fuzzy way, or a positive light, right? I mean, we think about these, detergents that we use to clean our clothes, and get them nice and clean. But we forget about the fact that a lot of these detergents still remain in measurable quantities in our clothes after we finish the wash cycle, especially in these high efficiency washers, which are so important. But we're really increasing our exposure to things like, again, like things like detergents. And we know that detergent application on the skin, even at some of these concentrations that we have in our clothes afterwards, really increases the how leaky our skin barrier is. And so I think number one, we spend a lot of time thinking about, like what are the microbiome effects, or what are the changes in our food. And I think all of those are really important. But when I think of the skin, I actually get more nervous about kind of what we're exposing ourselves to that's causing that leakiness in our skin barrier. And I've become increasingly more impressed with the research coming out, looking at the potential risk of having this long-term chronic exposure to detergents. And it comes from all of our day-to-day life. It comes from everything we apply to our skin.
Yeah, I mean, it's on your dishes after going through the dishwasher, right? And then you're putting your food on it and eating it. There's so many
Yeah.
things. And Dr. Andrew Wang had talked last month about these type of chemical exposures and
Mhm.
what we're doing for research in that area. But yes, I agree. Um, this one is from a scientist for sure. Nice talk, missed the early part. Does NK1 receptor play a role in SP induced DC migration? Or do human DCs express MRGPRX2?
So there are different, there's certainly expression of the MRGPRs on humans. So these MRGPRs, just for everybody in the audience, these are really interesting receptors that are expressed on a lot of different cells, immune cells, nerves, et cetera, that are really associated with itch and can be non-specifically triggered or specifically triggered by a lot of different things, from medications to all kinds of different triggers. So this MRGPRA1 is the mouse version, because obviously we did this research in a mouse model, but there is overlap between this MRGPR and human MRGPRs. Now, the question of whether or not the MRGPRs are expressed on human dendritic cells, it depends on the data set you look at, okay? Because sometimes it's hard to detect the level of expression of these guys, but it seems pretty clear that yes, that there is MRGPR expression on human dendritic cells. The question is, how much of that is inducing their migration? Does this totally apply with human in the human situation? We think it does, based on some experiments that we've done using human skin explants, but we don't have the full story on that. As for NK1, this is independent of that receptor.
Great. There's quite a few questions around like a carrier potential of allergen proteins with chemicals on our skin, and what chemicals are increasing in food allergies. How would you know this, and how would you, and how can others help?
Yeah, so this is really interesting. And I don't know any of the answers to this, but this is what gets me excited and like keeps me up at the middle of the night when my brain is just going, okay? But one thing that's always bothered me, right, is that, food allergens, they're boring. They're boring proteins, right? They don't do things, they're not enzymes, but we have these strong responses to them. And the immune system, when it comes to allergies, the immune system's a little sloppy with the way it does things. It doesn't always respond directly to the thing that activated it. It can respond to something that it sees in the same context as that activation stimulus. So that's always made me wonder, okay, well, what is the activation stimulus, right? And when you look at all of these food allergens, they're boring things like albumins and globulins and lipid transfer proteins. And, the more you read about them, the more you realize, okay, well, wait, lipid transfer proteins, they actually exist to move lipids around places, okay? Albumins and globulins, which I didn't appreciate, they are well known to carry small hormones and small molecules throughout our body, okay? And there's interesting work not done by many people. Nobody is doing this. This is why it's really exciting to be associated with FASI, where they're like willing to get involved with this this off the wall kind of research that nobody's thinking about doing, right? But there's some really interesting work where people are starting to catalog these small molecules that come along with some of these food allergens. And they are a specific type of molecule. They look a little bit like hormones, which is one of like the more frequent ones. And so we can track them and we can see them, and we can actually take them and then stimulate the neurons with them and see this activation. Now, a question is, okay, have they always been there? If they've always been there, why are we responding more to them? Well, the problem is that nobody's ever looked at them, okay? We don't know if they've always been there. We don't know how any of the ways that our farming techniques, or even potentially any of the chemicals that we're putting on our crops or putting in our livestock, okay, how any of that may be changing the concentration of these small molecules or even where they bind, okay? And this is crucially important, right? Because these are really, again, a black box of foods. It is amazing how little we know about these small molecules that come along in our foods when they make up doses that we would take for Lipitor or something like that. There are milligrams of these in our food that we're getting with each meal. So they clearly could be playing a role. And so one question again, how is this changing? How have we changed it with our farming techniques? Number two, what are they doing to us, right? We don't know those answers yet.
Yeah, no, it's fascinating, and this research is so interesting. And what I really like a little bit about what we're doing with the FOSSI and having you part of the group is that you're looking at one aspect in the skin of what it's doing to the skin, how it's
Mhm.
activating the neuroimmune. You're collaborating with Ruslan and Isaac, who are looking, and Romnick, who are looking in the gut, saying, what are these and Andrew, what are these chemicals doing in the gut? And what is that connection between the gut and the skin and the development
Right.
of these diseases, right? Whether it be asthma,
Right.
food allergies, or eczema. And it's really kind of that whole picture, because it is all connected and how,
Exactly.
and there's
Yeah,
nothing
because
worse than a chronic itch for a patient, right? And you
no,
can't help them. Like, I mean, I've had it, and it's
no.
just debilitating to them. So
I mean,
that's
it's like chronic itch is people kind of, I feel like like people don't really understand it. But we have all just with all with an acute itch, we've all experienced a bad mosquito bite that swells
yeah,
up or something like that, and we itch to the point of breaking our skin, right? We itch to the point of bleeding.
yeah.
And there are people that feel this over their entire body, right? And can you even imagine that? It
I
really,
can't.
it's absolutely intolerable. It's absolutely debilitating. And we need to have better therapies for folks.
Yeah, we really do, and I'm glad that you're working on it. So thank you and we didn't get to anyone's all their questions, but feel free to go to our website. Our email's on there, and you can email us if you have additional questions. But thank you so much for joining us today. Really exciting research. We're grateful to have you as well as as one of our investigators, and to be able to really partner with you. So thank you. FOSSI has single-handedly transformed how we diagnose, treat, and prevent food allergies, and we will not stop until we find a cure. Scientists and advocacy groups all over the world are watching our cutting-edge work, which is solely funded by generous donors. We need you to help power our next breakthrough. Donate now at foodallergyscience.org. 100% of your donation will go directly to funding research. And join us on our next webinar, Thursday, April 13th at noon, when you get to hear about Dr. Wayne Shuffler and his work. Until then, keep on imagining a world without food allergies, and FOSSI will keep building on it. Thank you. Thanks, Carrie.
Thank you.